Hello, everyone. This week we're going to talk about interactions between microbes and humans. Let's talk about our microbiome. First of all, what are other terms for microbiome? Some can refer to it as microbiome, some refer to it as microbiota, some refer to it as your normal flora. Those are all different terms for basically those microbes that are living on us, meaning like on your skin, for example, and inside of us, for example, in our guts. Now they could be in other locations. I'm just giving you like, two locations like the skin and the guts, but they could be found, of course, in different parts of our body. And those microbiome, they're actually part of us. So without them, we would actually get sick. Um, a recent study actually found that we have way more microbes compared to our human cells. So that research referred to humans as a bag of microbes walking because of how much microbes we have compared to our human cells, way more microbes. Um, and those microbes that are part of our normal flora, they're actually very important. They're critically important to our health. As I said, without them, we would be sick. Research has found that, some of these microbes can help you digest your food. So we would be at a huge disadvantage if we didn't have, these microbes in our guts. And they actually, did research and they found that individuals that are lacking some of these important microbes in their guts, they have gut problems. And so the doctors would try to go ahead and reestablish that microbiome for them in their gut. Um, some of these microbes, they can actually help you clot your blood. Okay, so when you get wounded, they would actually help you clot your blood. Some of them produce, certain, vitamins and, that we need. And so, therefore, these microbes, they are critically important to our health and to basically our, functioning. Okay, so without them, we would be in huge trouble. Now, as I said, these microbiome, our microbiota, they are basically our resident microbes. Those are permanent residents. And if you think about it, they have colonized us. They are growing on us, within us, and they've colonized, and basically, we humans are their home. And they've, again, like as I said, like they're part of us, and they're not causing disease. So if you're thinking about it, it's like we have way more microbes compared to human cells. How come we are not sick all the time? Well, these microbes, they do not cause disease unless something goes wrong, and we're gonna talk about it. But as long as your immune system is functioning properly, as long as you are not, becoming immunocompromised, where your immune system is, starting to weaken, etc., then you're definitely going to be okay. An infection happens when a microbe basically gets past your host's defense and enters your tissues and starts to multiply. And these microbes that will cause an infection, we're going to talk about them. Sometimes those are like total strangers kind of thing, like they're not part of your normal microbiota, and so they're kind of like invading your tissues and causing you to get sick. But actually sometimes there are these opportunistic microbes that are part of your normal microbiota, but they've entered into part of the body where they're not supposed to, and that can cause you sick, especially again if your immune system is weakened. So we'll talk more about that. And so, but let's say you do get infected and now you're not feeling well. Well, you're gonna you're gonna have a disease, and so a disease is basically like that term we use it when anytime you are not you're deviating away from your good health, and now you are at a state where your tissues are getting damaged. There is definite negative effect happening on your tissues, happening on your organs, etc. There's a project known as the Human Microbiome Project, and it's a huge project, and it started, since 2008. And this project actually, one of its main goals was to categorize the microbes living on, like, inside of us or on us. And it wanted to go ahead and identify these microbes, and also not just identify them, but see if there are any differences between my microbiome, for example, versus yours. They wanted to see what different individuals have similarities, how much differences there are. And of course, there are differences because a lot of things will determine what type of microbes you're going to have in your system. And, one thing would be the type of food that you eat, okay? And your lifestyle. And, like, if you're living alone, or if you're, like, sharing and living with someone else, because most likely if you're living with someone else and you're going to share food, drinks, etc., you're actually going to be sharing your microbiota with that person. So of course, our microbiota are not going to be identical. Um, but there's definitely similarities. So again, this project kind of looks at all of that stuff, and they go ahead and they use, like, genomic sequencing so that they can go ahead and tell exactly, the type of, bacteria we have by doing, like, genetic sequencing. And so it's a huge project known as the Human Microbiome Project, and they've discovered actually so much about our microbiome. They've also discovered like parts of your body where there is that microbiome. So parts of your body where they thought previously there shouldn't be any microbes in it, and then yet went ahead and they found some. So one of those parts of your body would be the lungs, where for the longest time we thought our lungs are those, like, sterile, it's that sterile environment. No microbes are found in the lungs, and it turned out not to be true. They actually did find microbes within our lungs. And when they compared it actually to the microbes in our guts, well, it is different. Okay, so they kind of said that, also different parts of your body could have its own microbiota. It doesn't always have to be exactly identical within your own body. So that was a very huge, like, discovery that they also, found out. Now these are actually, like, some of the sites that we kind of previously known that there are normal microbiota. And again, with the Human Microbiome Project, they did kind of confirm that, yes, indeed, those are the places where we do have, microbes, and that's definitely, like, our skin, our mucous membranes, for example, like the female vagina, the ears, even your eyelids, your mouth, of course, your GI tract, your urethra, where you void your urine. And then some additional sites that they went ahead and they figured out, okay, it does have normal microbiota, the lungs, and as I said, like, for the longest time, they thought the lungs are like a sterile place, the bladder, and so therefore, the urine. So again, the bladder for the longest time, again, thought that no microbes are there, but actually recently they found out, nope, it does have also some. The breast and breast milk, so a newborn, that first batch of milk that they would get, uh, there we go, they are getting some microbes, and from the breast milk. Um, even the, like, even the, like, when the baby or when the fetus is in, the uterus and in the mom's, like, placenta, they actually found out there are also microbes there. And so the fetus is already collecting microbes even before, getting born. And of course, as as the mom goes ahead and gives birth to her child, and they pass through, the vagina, they're also going to pick up, like if it's a natural birth, they're also going to pick up bacteria. And even the amniotic fluid. So as I was saying, like, so the amniotic fluid does have, like, they found out that it does have microbiota. And then parts that, okay, don't have microbes, but they detected like the DNA, okay? So let's say a microbe kind of like, burst and its DNA, got released. They actually found some DNA of the microbes in the brain and in the bloodstream. Again, not the microbe itself, okay? It's a huge difference, but, they found remnants of it, like parts of its DNA. Now, our human body, of course, like, does have variations, like, in temperature, pH, nutrients, oxygen, meaning that not all parts of your body are going to have exactly the same pH, for example. Even like, for example, if we think of our blood, when you take a look at the blood is around like a pH, like that's neutral, right? Like in the sevens, but if you compare the pH of your stomach, it's a different pH. And so therefore, within your body itself, we have different regions that have, different, pHs, etc. Oxygen, for example, like the oxygen is going to be high in your bloodstream, but if you take a look at the oxygen levels in your colon, it's really, really like low. And so that means that different microbes are going to inhabit different parts of your body, right? You're not going to find, the same microbes that are living like in your stomach. You're not going to find them the same as the ones living in your colon, because the pH is completely different, right? The pH in your colon, again, is not acidic compared to the pH in your stomach. It's highly acidic. So different parts of your body are going to have different microbes. And they actually found out that, these normal biota that we have, they're going to be kind of living together in close proximity, so they kind of become a community, okay? So these microbes, they become a community, and it's not so easy to go ahead and displace them by new incoming microbes, okay? So new incoming microbes have a hard time actually, establishing themselves amongst the normal microbiota, because the normal microbiota, um, they kind of resist, okay? They resist that change. Like, you're a newcomer, well, it's not going to be so easy for us to go ahead and accept you. And they found out that, like, your normal microbiota could actually be producing like certain chemicals, etc., that would actually make it hostile to new incoming microbes, okay? So therefore, like, new microbes incoming, would have a hard time becoming part of, the microbiota. And I mean, if you think about it, that could be like a pretty good thing, especially if, let's say, this new incoming microbe is a pathogenic one and it can cause harm. Well, it's not going to be then super easy for that microbe to go ahead and kind of colonize, because the normal microbiota is, protecting us. And so, which kind of brings us back to the discussion of the antibiotics that we've discussed last week. And we were saying how, excessive use of antibiotics and excessive use of broad spectrum antibiotics that can kill a whole lot of your normal microbiota can cause, problems, because you are breaking down that normal microbiota that could be protecting you from these, disease causing microbes from colonizing and taking over. Now, what could be some factors, though, that can weaken your immune system, and so therefore would put you at more risk of catching an infection and, becoming sick? Because if you think about it, right, microbes are ubiquitous. They are found everywhere. Yet we do not fall sick, pretty easily, or, we don't fall sick like all the time. And you could think, you should thank your immune system for that, trying to protect you and everything. And as I said, even your normal microbiota, they are also resisting, the new incoming that could also be pathogenic. But I mean, we do fall sick, right? That's just a fact. And so, what could be some reasons? Well, I mean, and are there individuals that are more prone to getting more sick compared to others? Of course. One is age. So, like, one factor is your age. So, the older you get, unfortunately, your immune system starts to get lower and lower. And that's something we're all gonna face as we grow older. Our immune system is just not gonna work as good. And then another thing, if you are extremely young, like if you are an infant, like a newborn, etc., then again, your immune system is still pretty weak. Um, Genetic defects in your immune system. So some people do not produce like enough white blood cells, etc. So they have a genetic defect and their immune system is already pretty low, or acquired defects. For example, someone acquired AIDS, the HIV virus, which definitely um, lowers their immune system, so these individuals would be immunocompromised. Certain, actually certain diseases like individuals that have the diabetes, also their immune system would be much lower. If someone had surgery, again, surgery will go ahead and lower your immune system. Organ transplants. So when someone, by the way, is getting an organ transplant, they go ahead and they lower your immune system intentionally because they don't want you to uh, reject that organ that they are about to give you. So they intentionally lower your immune system, which is kind of dangerous, but at the same time, definitely necessary. But, it's definitely necessary because they don't want you to reject the organ, but at the same time, it's dangerous because you then are more prone to catching an infection at the hospital. Um, Things like cancer, would lower your immune system, and especially, with the chemotherapy drugs that someone would take, it would actually lower your immune system because a lot of these drugs would actually kill your white blood cells. So your immune system takes a huge hit. Um, and then, as I said, like things like diabetes, liver malfunction, etc., chemotherapy, immunosuppressive drugs, and some individuals, have to take those immunosuppressive drugs, for a pretty long time, if not like, forever. So again, some people have to keep taking those immunosuppressive drugs. Let's say they just got an organ, they don't want it to be, rejected. They would have to take those immunosuppressive drugs, for long terms. Um, and then stress, so try not to stress out too much. I know it's kind of like, hard. And there are situations where you're going to stress about them, but chronic stress is not good. Okay, so continuous stress is not good. We all get stressed one point, several times in our lifetime, to be honest. But, what I'm talking about is chronic stress. Continuous stress is not good. It does lower your immune system. Definitely pregnancy, because your immune system is going to decrease when someone is pregnant because your body, if you think about it, I mean, kind of, it's good because you don't want to be attacking your own baby, right? Because now the baby's going to be like a foreign thing growing, right, in your uterus, and so your body, again, like your body is going to see that as something foreign that, okay, needs to be attacked. So naturally, like your immune system lowers to prevent that. But I mean, it's good because you're not going to be, harming your own baby, but at the same time, like the pregnant female is more prone to then getting infections. Um, And then, if you are fighting off like an infection in the first place, then again, your immune system is going to lower. Where do babies get their microbiome from? Well, as I said, like in utero, okay? So when they are still in the placenta, they actually found that there are microbiomes in the, like, within the amniotic fluid and everything. And so, um, as I said, they thought previously that this is a sterile location, but it turns out not to be true, and there are microbiomes, and so we, like in the womb, the baby is already, picking up microbiota. And then of course, like, when labor is going to take place, now they actually found out that vaginal versus C-section births will contribute different microbiota to the baby. So a baby getting delivered naturally and passing through the vagina will go ahead and pick up the microbiota within the vagina. And the vagina, by the way, has a lot of lactobacillus. So they found out, like, the baby will pick up a lot of lactobacillus, like, along the way. But they found out that, okay, what if, what if the female went and had a C-section? So the baby's not going to be passing through the vagina, and they're not going to be picking up those microbes, right, in the vagina. And so they actually found out a huge difference between the microbiomes of those that are born naturally versus those that are born via a C-section. The milk, so the breast milk, does have microbes. And so again, if a baby is going to be fed breast milk versus the formula, then again, they are going to have different microbes. And they actually, like, there was another research that found out that infants that do get breast milk, because it does have microbiomes, and it turns out breast milk also has antibodies. They found out that those babies tend to do better compared to those who are, purely on the formula. But they've done a lot of like work with the formula to try to make it like mimic, the breast milk. Um, Caregivers, right? So, I mean, when you go ahead, let's say, kiss your baby, there you go. Like, you just went ahead and actually transferred some of your bacteria, that's in your saliva to your baby. So again, siblings, etc., they're definitely gonna share, their microbiome unintentionally, with the baby. And so therefore, though, you have to be very careful, though, especially with like newborns, um, because their immune system is not that great. So you have to be very careful, if you are carrying, like, let's say, um, um, a cold sore. Let's say someone has a cold sore, okay, which is caused by a virus. You do not want, to kiss the baby when you have a cold sore, because, again, the immune system of the baby is not that great, so they're actually going to have a very hard time, and it could be fatal for them to fight off such an infection, okay? And then the environment, so that's why I was saying, like, depending on where you're living, you could actually be picking up different microbiomes compared to, if someone is living somewhere else. So even the environment that you're in is going to also kind of determine the microbes that you're going to be picking up, and, things that you are touching and getting in contact with, etc. And then of course, like, the food, okay, as the baby grows older, the type of food that they're going to be consuming, of course, all of this is going to contribute, to their microbiota. Now, a pathogen, as I said, when we talk about pathogenic, if we're talking about pathogenic microbes, well, these are microbes that are going to go ahead and result basically in infection, and they will cause disease and they will harm you. Now, so when we talk about pathogenic bacteria, okay, there are true pathogens and there are opportunistic pathogens. This is kind of what I was talking early on when I was telling you, there are those opportunistic pathogens that you would get sick and by these opportunistic pathogens that are actually part of, they could actually be part of your normal microbiota, but because your immune system, let's say, took a hit and your immune system is now pretty low, these bacteria that were not causing you harm back then could actually do cause you harm now because your immune system is now much, much lower. Or if these pathogens kind of like, get established in part of your body, uh, where they're not supposed to be there. So for example, if you have a staphylococcus, right, on your skin, and you go ahead and you cut yourself, okay, pretty badly, and some of that staphylococcus now entered into your bloodstream where it's not supposed to be, right? It's supposed to be just on your skin. Its home is not supposed to be the bloodstream. That then could actually cause huge problems and it will become an opportunistic pathogen because now it's in a part that's not natural to them and they will start causing uh, pretty bad things to you. Or as I said, some of these microbes, living within you, they're not making you sick, but all of a sudden, if your immune system, is now lowered, those bacteria could go ahead and start kind of, harming you. So these are called opportunistic pathogens. True pathogens, those are, let's say, for example, a bacteria that will cause disease in a healthy individual. So it doesn't have to do with like, oh, your immune system is low or whatever. Nope. If you've ingested, let's say, a lot of these pathogenic bacteria and you got exposed to a high load of them, and they will go ahead and harm you, even if you have a normal immune defense, right? So I mean, um, even if your immune defense is good and it's not really immunocompromised or anything, how come you still get sick? Well, because you did get exposed to, a certain dose of that pathogenic, bacteria. Now, it turns out actually that the dosage differ from one pathogenic bacteria to another. So depending on the bacteria we're talking about, some bacteria, if you get exposed to just like a single dose of it, that's it. That could make you sick. Some of it, you would actually need like a higher dose for you to get sick. So there are multiple factors that would also kind of determine, why someone would get sick versus someone else wouldn't get sick. It depends, okay, how much dosage, right, of that pathogenic bacteria were they both exposed to, and whether the bacteria, like, you were exposed to it and it did enter into your body. Again, all of these factors will determine whether you're going to get sick or not. But that's kind of the difference between true pathogens, though, and opportunistic pathogens, okay? Now, when we use the term virulence, we're basically talking about how, basically the severity of the disease caused by, again, a certain microbe. So how severe, okay, is it going to make you sick? Like the degree of how pathogenic it is. And it differs, okay, from one microbe to another. Some microbes have a pretty high virulence, and so they're going to make you very, very sick. And then some microbes, even though you're infected with them and everything, well, they're not going to make you that sick. So it depends actually on the microbe. And it depends on, the microbe itself. It depends on how it's going to establish itself within you. Is it going to like multiply and colonize, pretty fast, kind of take over? Is it like, how much damage is it causing? Okay. Is it like killing a lot of your cells? So the degree of damage that it's causing to you will definitely, make a huge difference in terms of the virulence of microbe A versus microbe B. You hear of individuals that, get sick with a certain microbe, and I mean, within a few, within a week or so, I mean, they're feeling better. Versus someone, getting sick with, let's say, a different microbe, and it's a totally different situation, and they are, at the hospital, and, it could actually be fatal. So it depends on the microbe itself and how much damage it's causing, and how fast, for example, it's multiplying within you. These are certain factors that would make, one microbe like pretty virulent versus another one that's not. And it also has to do with the characteristics of the microbe itself. Like, do they have characteristics? Like, do they carry, for example, a capsule? If you guys remember, we talked about capsules before. Those are like those hard outer shells. So bacteria, for example, that are encapsulated and they do have those capsules, it makes it super hard for your immune system to actually fight them off. So then those encapsulated bacteria, well, then they can have, more advantage in terms of colonizing, and taking over. And so, again, like, certain characteristics or like structures that they have, okay, will make a difference. What this table is showing you right here is how the bacteria enters into our body. So they can enter through our skin, or they can enter into our GI tract, into our respiratory tract, or into our urogenital tract. So, for example, how are they going to pass through our skin? Well, if you get cut, right? So cuts and nicks, abrasions, all of that, okay? Areas basically of broken skin, they can go ahead and get into it. Examples like Staphylococcus aureus, Streptococcus pyogenes, and that's why, a lot of the antibiotics that you can apply to your skin would go ahead and target, like, Staphylococcus and Streptococcus, because those are highly common, on the skin. They would enter through it. So, or, they enter through like the mucous membranes, for example, of your lips. Okay, so a lot of the cold sores, etc., they would actually be to the, like, herpes viruses, and even worms. Like, we're not just talking about bacteria here. We're talking about bacteria, viruses, like worms. They can they can burrow themselves, um, basically like penetrate through your skin. So that's why it's not recommended at all that you would be barefoot and put your feet in a lake, because it could have like worms, and without you noticing, without you feeling it even, it can go ahead and penetrate through your skin. And a lot of, like, the infections with worms actually happen like that way. Insect bites. So if the insect is carrying the microbe, then and the insect goes ahead and bites you, that little bite, there we go, like, it goes ahead and injects the the microbe inside of you. So a lot of viruses are passed that way, even bacteria, etc. So, for example, like the West Nile virus, malaria, etc., could be passed that way. And so, a lot of times, like, to prevent that, what they try to target, if they want to get rid of like West Nile virus, is to get rid of the mosquitoes, right? Because mosquitoes um, carry the West Nile virus. So if we go ahead and eradicate the mosquitoes, then you can eradicate the West Nile virus. And then they can even enter it through your eyes. So they can, like, penetrate through the layer, like, in front of like your lens, like your conjunctiva. Or it's not like in front of your lens, it's like the part that's basically covering the if you guys recall, like your anatomy, your sclera, that white part of your eye, we have a layer called conjunctiva, and it can go ahead and infect that. So pink eye, for example, is caused, it's an infection to the conjunctiva of your eye. Um, it can enter, like, microbes can enter through your, like, into your GI tract. How? Well, when you eat or drink contaminated food and fluids, basically, you go ahead and you ingest it, and there we go. It's now in your GI tract. Or let's say you touched your computer, and then you went ahead and you put your hands in your mouth. There you go. So, this is how it can enter into your GI system. A pretty famous one that would inhabit like your GI system, for example, like E. coli, and that's why you hear a lot of like outbreaks, for example, with things like lettuce or spinach. There's an outbreak of E. coli, there's an outbreak of salmonella, and so, lots of cases of E. coli infections and salmonella happen, because you've ingested, food that was contaminated with these, type of microbes. We can even inhale them. Okay, so you can even inhale them. So a lot of the influenza viruses, the common cold, etc., those are actually like, you can go ahead and inhale them, and that's how they can make you sick. Streptococcus pneumonia, you know, um, like, Mycobacterium tuberculosis, the one that causes TB, these again could be inhaled. And then some of them enter through the, like, into the urinary tract. So basically, like enters through the skin of the reproductive organs, like, the penis and the vagina, etc. So a lot of these you would find that, syphilis, etc., Neisseria, gonorrhea. So a lot of these would be like the STDs, right? The STDs type of infections. Um, that can, again, all it takes is a simple like cut or abrasion, and they go ahead and they pass through that broken skin. Now, the quantity of the bacteria that will make you sick, we refer to it as the infectious dose. And the infectious dose is basically the minimum amount of microbes necessary to go ahead and cause an infection. And that infectious dose will differ from one microbe to another. So some microorganisms, they will make you sick, and they will go ahead and infect you with a very small infectious dose. And so these would mean that they have a pretty high virulence. For them to make you sick, even though you were exposed to a very low amount, that means that these bacteria, I mean, these are pretty serious, type of microbes, because they have great virulence. So as you can see, like, infectious disease, like infectious dose for the Q fever is just a single cell. That's it. That's all it takes, and the person would go ahead and get sick. Versus cholera, for example, it would take a whole lot, right? So that's like a billion. Typhoid fever, it's like 10,000 cells. Gonorrhea is about 1,000 cells. So um, um, as you can see, definitely there is, like, differences between the infectious doses of the bacteria. Of course, the smaller the infectious dose is, the more virulent that microbe is. Now, once the bacteria enters into the host, the bacteria or the microbe in general, okay, again, it could be a bacteria, it could be a virus, it could be a protozoa, worms. Once those microbes have entered into the host cell, they need to adhere to the host's tissues. And that's because the host is going to try to, flush them out, etc. And I mean, there are so many different mechanisms, to try to, flush these microbes out of your system. And so a firm attachment of these microbes to your tissues is really essential, so that they can, cause disease. And so um, the binding between the microbe and the tissue varies. Some are going to go ahead and bind, because they have specific proteins, and they're going to bind to specific receptors. For example, on the tissue. If we're talking about like, worms, well, I mean, they could have the hooks and the suckers that they're going to go ahead and grab onto the tissue that way. So, I mean, there are different mechanisms of how they're going to attach to the host cell, but at the end of the day, they really have to attach. They really have to attach to the host tissue so that they can go ahead and start causing disease. And some of them are very specific in where they can bind. So some of them are very limited to the cells that they can go ahead and bind to. Because again, depending on the receptors that they have, they can, let's say, for example, only bind to the tissues in the colon versus they can only bind to the tissues in, the respiratory tract. And so therefore, again, some of them are very limited to the cells that they can go ahead and bind to. Now, some examples, okay, of how they're going to attach to the host tissue. Some of them have these fimbriae. For example, the bacteria that have those fimbriae, they're kind of like little tiny hair-like structures that helps them actually stick to the tissues. Or they can have some specific proteins on their surface that will go ahead and bind to specific receptors. Or they can have this like slime layer that they have around their cells. So again, like, some bacteria definitely have those capsules or those like adhesive, like, slime layer that would help them go ahead and attach to the host. Viruses, for example, they have specific proteins on their surface, and so they will bind to specific receptors of the host. Parasitic worms, as we just said, they will go ahead and use their suckers and hooks to go ahead and latch on to the host tissue. Now, your immune system is definitely going to try to go ahead and attack, right, these microbes, okay? And so part of our immune system, part of our white blood cells, we have these phagocytes, and these phagocytes, they basically eat up foreign particles. So they go ahead and engulf, and they try to destroy basically these pathogens. How do they do it? Well, first they swallow them up, and then inside of the phagocyte itself, there are a bunch of enzymes. And there are a bunch of antimicrobial chemicals that goes ahead and basically like breaks down the microbe inside of the phagocyte. But there are, some microbes that will actually, resist being engulfed by the phagocytes. So some pathogens, they go ahead and they release, for example, certain enzymes that will actually break down the phagocytes. So some of these enzymes are known as leukocidins, and those are basically, chemicals that are produced by, the microbe, and it can go ahead and actually kill the phagocyte right away. So this is how they can evade the phagocytes. And then depending on whether the bacteria, for example, has like a slime layer or a capsule, right? This will make it hard for the phagocytes to actually engulf them, okay? And then it turns out that there are some bacteria that even though they got swallowed and they got engulfed by the phagocyte, guess what? They can still survive inside of the phagocyte. So even though, they have now been ingested by that phagocyte, they can still survive. Those enzymes that the phagocyte has, and what will happen is, well, they will go ahead and start replicating within the phagocyte, and so that will give them definitely a chance, to go ahead and start spreading around. If they burst out of that phagocyte, then they can go ahead and start spreading, because even though the phagocytes have ingested them, it cannot kill them. So when we talk about microbes, we said that some of them are highly virulent and some of them are not. And so we said that those that are highly virulent, they're going to cause a whole lot of extensive damage to the host tissue pretty fast. So if we think about, okay, how do these microbes, really affect the host in such a negative way? What are they producing that would affect the host in a negative way? Well, some of them produce enzymes. And these enzymes are pretty powerful, okay? And we're going to discuss a few of them in destroying the host tissue. Some of them produce toxins. And there are two types of toxins that we're going to discuss, endotoxins and exotoxins. Some microbes out there, what they do is they go ahead and they induce the host to actually attack them. But when the host goes ahead and attacks them, your host, your body, goes overboard and responds in an excessive, manner, to the point that your own immune system is really going to be harming you. So, for example, your immune system can launch an extensive inflammatory response. And so, basically, like, inflammation will take place like within your whole entire body, and that can definitely affect you in a very negative way. This is just like, an example. And so instead of the host kind of like helping you, I mean, it's actually going to make things worse because you are responding in an excessive, manner. Um, and so sometimes you would hear that actually, this individual, unfortunately, had an infection and they died because their immune system, not because it wasn't actually helping them out or because their immune system was low. No, on the other hand, it was the opposite. It was that their immune system launched an extreme attack to the point that your tissues themselves got affected in a negative way, and it can cause fatality. Some microbes, they actually change your DNA. So they can actually cause, for example, some of them can cause cancer. So they can go ahead and mess up your genetic, information, and they can cause, and that can lead to cancer, for example. Um, and so you would hear, of, certain viruses and their potential of causing cancer. And so that's because, again, they can have the ability to go ahead and change the genetic information of the host. So right here, you guys can see, a bunch of images of how a microbe can damage the host cell. So in this image right here, you guys are seeing that this bacteria is producing certain enzymes. And those enzymes, we're going to go ahead and actually discuss a few of them. But you can see from this image right here that those enzymes are actually breaking down uh, what's holding the cells together. So this is how then it will start kind of breaking down like your tissues, and it can actually start allowing the bacteria to even spread further, okay, because it's kind of like breaking that barrier, right? So let's take a look at a few of these enzymes. So let's take a look at exoenzymes. Exoenzymes, those are enzymes produced by the bacteria to basically inflict damage on the host tissue. And what some of these enzymes do is they go ahead and they break down the tissues, and therefore when they break down the tissue, how's that going to help them? Well, they're going to be able to spread throughout your body. And so if you recall just the image that I showed you earlier of our cells are usually lined up one next to the other pretty tightly. So what happens is they go ahead and they break down what's holding your cells together. And by doing so, again, they can help spread throughout your tissues. So an example would be mucinase. That's an enzyme that will break down that protective layer that we have on our mucous membranes, and again, that will help them spread. Um, hyaluronidase, and that's another enzyme that will break down basically what holds your cells together. And so again, that will help them spread. Actually, some drugs that we take would have this enzyme within it. And the reason why sometimes they add this enzyme to the drugs that we take to actually allow your body to absorb that drug faster, and so that this drug can actually spread throughout your body pretty fast as well. So some drugs actually add this enzyme within it. There's another enzyme called coagulase, and it will cause your blood to go ahead and start clotting. Um, Again, for us as a host, that's not a good thing. That's definitely going to harm us. So again, some bacteria go ahead and do that. Some bacteria, they do the opposite. Instead of clotting your blood, they go ahead and they do the opposite if they release this enzyme called kinase. Kinase does is it basically breaks down the clots. Why would they want to break down the clots? Well, because again, they want to go ahead and be able to spread faster. So some of them do clot your blood, some of them actually do the opposite so that they can spread faster. So these are some examples of exoenzymes. Now, another way microbes can damage the host is they release toxins. And we're going to talk about two types of toxins. There's exotoxin and there's endotoxin. So let's go ahead and take a look at the toxins. A toxin basically is just a product, right, that the microbe is producing, and that toxin is basically poisonous, right, to the host. And toxins are usually, when we come and name them, we name them according to what are they targeting. So a neurotoxin is going to affect your nervous system. Enterotoxin is going to affect your intestines, and that's why we call them enteric bacteria, right? The ones that live in your intestines, because entero means like intestines. Hemo coming from blood, so those are toxins that will break down the red blood cells. Nephrotoxins coming from the word nephron, so those are going to damage the kidneys. Now, the hemolysin, those are basically exotoxins produced by the bacteria, and what these hemolysins exotoxins do, they're basically going to break down the cell membrane of the red blood cells, causing, the red blood cell to burst. Now, this is a blood agar plate right here. And what happens is what you are seeing right here are three different types of hemolysis taking place. Now, this one right here is called gamma, okay? So this one right here is gamma hemolysis, and gamma hemolysis means that this bacteria right here that you just inoculated on the blood agar plate, which, by the way, this plate does have red blood cells, and we usually add sheep red blood cells. So this plate right here has sheep red blood cells, and let's say you went ahead and you separated this plate into three different quadrants. On one quadrant, on each quadrant, you basically inoculated your bacteria. This one right here, this is called gamma hemolysis. This means that the bacteria is not capable of breaking down the red blood cells, okay? This one right here, this is beta hemolysis. This is a complete breakdown of the red blood cells happening. This one right here, when you see it kind of greenish yellow, this is partial hemolysis, and we refer to it as alpha hemolysis. So alpha hemolysis, that's when the bacteria is partially breaking down the red blood cells. So you have gamma hemolysis, no breaking down of the red blood cells is taking place. Beta hemolysis, complete breakdown of the red blood cells. Alpha hemolysis, partial breakdown of the red blood cells. And the reason why the bacteria, by the way, would want to break down the red blood cells, it's because they are after, the component inside of the red blood cell, which is the mineral that we have inside of that red blood cell, which is basically the iron. So that's why they would go ahead and, break down the red blood cells. But again, for them to do so, they would have to release those toxins, the hemolysins, to go ahead and break down the red blood cells. And when the red blood cells burst open, of course, the hemoglobin protein inside of it, it's going to go ahead and get released. And by the way, studies have found out that when the hemoglobin protein is within your red blood cells, that's fine. But if that hemoglobin actually bursts out of our red blood cells, this is toxic for us humans, okay? So that's another thing to consider. Now, when we refer to endotoxins, endotoxin is basically a single substance, and it's within the liposaccharides. It's basically the liposaccharide part within the gram-negative bacteria, in the outer membrane of gram-negative bacteria. So all gram-negative bacteria, by the way, are going to release the endotoxin. And these endotoxins, they can go ahead and affect, having a systemic effect on the tissues and on the organs. They can cause fever, inflammation, hemorrhage, and diarrhea. And usually infections by Salmonella, Shigella, Neisseria, meningitis, E. coli, those are pretty dangerous, and they release, so all of these are gram, negative bacteria, and they are pretty dangerous because when they release that endotoxin, your body can actually go into shock. So gram-negative bacteria, they're going to shed that lipopolysaccharide into the tissue and into the circulation. And if you recall, when we were talking about gram-negative bacteria earlier, we were saying that they have that lipid A, right, which is that endotoxin that they have. And so when they go ahead, shed it into the system, we can definitely have pretty nasty side effects. And again, the person can go into shock and it could be fatal. Now, those endotoxins, usually like the bacteria would go ahead and release them when the bacteria is also like dying. So as the bacteria dies, these endotoxins are going to go ahead and get released. So even like if your body is trying to get rid of that microbe and it's trying to go ahead and kill it, I mean, it's still pretty bad because as it's dying, it's going to go ahead and release that endotoxin into the tissues. This table right here summarizes the differences between the exotoxins and the endotoxins produced by bacteria. So let's go ahead and take a look at the differences. So in terms of toxicity, the endotoxins, you would need higher doses of the endotoxins for them to really be toxic versus the exotoxins, very small amounts, and they are highly toxic. So exotoxins, they are way more toxic compared to endotoxins. Exotoxins can be produced by both gram-positive and gram-negative bacteria, while endotoxins, they are produced by gram-negative. So gram-positive bacteria will not produce endotoxins because endotoxins, they are part of that outer membrane of the gram-negatives, right? They are part of that lipopolysaccharide, and so therefore only gram-negatives will produce the endotoxins. Exotoxins, as I said, can be produced by both gram-positive and gram-negative, and they're basically proteins that the bacteria goes ahead and releases. In terms of the effects of these toxins on the body, endotoxins, they're systemic. So they can spread basically like everywhere, and they can cause fever, inflammation. The person can actually, go into like a shock. Exotoxins, they're very specific. So they only target like specific cells. So for example, like they would only target the liver cells, or the neurons, or the lungs. So they're very specific in what they are targeting. Endotoxins can actually withstand, heat such as like 60 degrees Celsius, whereas the exotoxins, they're not very stable, okay, with heat, so they can break down. In terms of toxoid formation, so what is toxoid? Toxoid is basically an inactivated toxin. And exotoxins can be converted to toxoids. So they go ahead, they take the exotoxins, they um, expose them to certain chemicals, okay, like iodine, for example, bleach, etc. And what these chemicals would do is it would go ahead and inactivate the exotoxin. Then they will take that inactivated exotoxin, which is now you can refer to it as toxoid, and they can actually use it as a vaccine. Okay, so they would inject someone with these toxoids, and even though they are inactivated, so they're not going to make us sick, but your system can go ahead and start producing antibodies against it. Okay. And so, therefore, your body can go ahead and start producing these antitoxins, which are basically those antibodies against it. Now, endotoxins, we cannot do that. So, endotoxins doesn't work, and we cannot, convert them to toxoids, and therefore, we can't use them, in vaccines or any of that. Okay, so that is specifically, only for exotoxins. Now, in terms of fever stimulation, whether they're going to cause fever or not, endotoxins, yes, they do. Exotoxins, like usually not. I mean, they can, but again, usually not. But with endotoxins, yes, definitely fever. Um, and then, in terms of, how do they get like, released? Well, they can definitely, endotoxins, when the bacteria itself breaks down, that's when, or lysis, that's when it goes ahead and it starts releasing the endotoxins into the body. Exotoxins, I mean, as I said, like they have very specific targets, and then, the bacteria goes ahead and releases these exotoxins. So they're going to be released from, those live bacteria cells, and again, they're going to target specific, targets in the body. Um, and then, as I said, gram negatives, all gram negatives, by the way, are going to have the ability to produce endotoxins because, as I said, they're, it's part of their lipopolysaccharide, right? Part of that outer membrane that they have. Exotoxins, gram positives and gram negatives can go ahead and produce exotoxins. And actually, exotoxins are different, right? So you can have an exotoxin that would target the red blood cells and cause hemolysis of red blood cells. You have an exotoxin that would target neurons. You have an exotoxin that could target, liver cells, etc. So exotoxins, they are different. And one bacteria can actually produce several exotoxins. Um, endotoxins, the endotoxins are all the same, right? So the endotoxin, it is the same, and as I said, they are produced by the gram-negative bacteria. Um, Now, another, thing to consider is let's kind of, let me give you a few examples of the exotoxins. So, for example, the botulism, okay, disease. It's actually caused, by a neurotoxin, which is an exotoxin that's going to go ahead and cause it's going to be transmitted to, like, it targets like the nerves, and so it causes, paralysis, tetanus, the tetanus disease. So again, the bacteria Clostridium tetani goes ahead and also produces that neurotoxin, which is going to cause your muscles to contract uncontrollably. So that's again another example of exotoxin. So um, these are some, like, examples, okay, of exotoxins. And so this kind of, like, right here summarizes, you guys, the differences. Make sure that you know the differences between exotoxins and endotoxins. Some of the terms that we use when someone has a disease, so those are kind of like warning signals, signs and symptoms and syndromes. So a sign is basically an objective evidence, like you, the observer, can see or you've measured something, and so you know that this is a sign of this disease. So for example, you've taken a sample of their blood and you found that there's bacteria in it, okay, which is a very bad sign. So that's a sign, right? You're able to see that there's bacteria in their blood or, in another sample, okay? You see that there's bacteria in it, or you went ahead and you did a blood test and you can tell that their white blood cells, it's pretty high, so kind of indication that they're fighting off something. So that's a sign, right? High numbers of white blood cells, there's bacteria in the sample, etc. Okay. A symptom is subjective evidence, so it's what's sensed by the patient, right? So they sense, like, pain. Um, and of course, you have to kind of, believe them, right? If they tell you, like, I feel pain 10 out of 10, even though they don't look, that they are in pain to you, I mean, do you believe them, right? This is what they are sensing. They are sensing that this is how much pain, right, that they are in. Or if they tell you, well, I feel like I don't feel right. So it's kind of like feelings, right? Sensations of what they are, what they're feeling, etc. Or if they tell you, well, I feel like I'm bloated or I'm swollen. Now with swelling, you can also see it as a sign because you will see the edema. And so you can definitely, swelling can be both like a sign and a symptom. Fever are really the same because, I mean, they can feel hot, right? So I feel like I have a fever. And yes, you can go ahead and measure the temperature and, again, it's a sign they do have a fever. Um, a syndrome is basically when you have like a complex, right, of like signs and symptoms. And we go ahead and say, okay, it's a syndrome, and based on that, complex of signs and symptoms, you can identify, or identify the disease. Of course, you have to be you do tests and everything so that you can determine whether they have this disease or not. But again, like, there are lots of diseases out there that that it's going to cause the following symptoms, it's going to cause the following signs, so it's going to be easy for you to identify the disease. And then this is just, like, again, like a few examples. So like symptoms, someone might feel, again, like they're hot, like they have a fever. And again, you can actually get a measurable number, right, when you check their temperature. Now pain, you can't really measure that. So again, this is like definitely a symptom. Or when they feel sore, again, how are you going to measure that really? Swelling, okay. But with swelling, as I said, I mean, you can go ahead and measure it, or you can see it, right, as an observer. Like, you will see that there is edema, there is accumulation of fluid. You can totally tell, let's say their legs are swollen, okay. So granuloma and abscess. So like when again, you can see that, okay, when especially like they do scans, etc. You will be able to go ahead and see the abscess and see, basically, abscess, you guys, is like walled off collection of some white blood cells and some microbes. And abscess is never a good sign, okay? Abscess is not a cyst, by the way, okay? So an abscess, you definitely have microbes in it. And so it will kind of, they might refer to it as like pus, okay? So you might heard like, oh, this individual has an abscess and it's filled with pus. Pus is basically a bunch of like white blood cells along with microbes. And it will have to be removed, right? Because you don't want that abscess to be within someone's body, because what if it breaks off? Because it is walled off, but if it bursts, then it can cause a huge problem and the person can go into a septic shock, because it can go ahead and start spreading basically to your tissues and to the bloodstream. It would be a major disaster. Lymphadenitis, that's another kind of like, sign. I mean, the lymph nodes are going to be swollen. And the common places, by the way, for someone to get a swollen lymph node if they have infections is the lymph nodes in your neck will swell, the lymph nodes under your armpits, and the third location where we have lots of lymph nodes, and you might notice swelling there too, is near the groin area. So those are That doesn't mean that other parts of your body don't have lymph nodes, but those are the three major parts that do. Um, Signs of infection, as I said, if you take a sample of the blood, and you see that there's an increase in the white blood cell, which we refer to it as leukocytosis. Leukopenia, that's when you notice that the white blood cells are very low. Septicemia, that's when basically it's kind of like a general state where the microorganisms are multiplying in the blood and they're present in such high numbers. Again, this is a very bad sign. Bacteremia, basically like the viruses are high in number and they're present in the blood, but they're not multiplying, okay? Viremia, again, viruses are present in the blood, but again, not really, multiplying. So these are some signs, again, they take the blood sample and they can go ahead and take a look at it under the microscope and, and see, if there is an infection or not. And then they can, take a look at the blood sample, like, at different intervals and see whether the infection is getting worse or whether it's going to stable. Okay, and so this is how they would know whether, again, the microorganisms are really multiplying in your blood and things are getting out of control, or, if it's kind of like stable and they're not multiplying in your blood. And so there are infections that do go unnoticed, unfortunately, because some do not even cause any symptoms, right? So you are carrying the microbe and you could actually be shedding it, okay, to others without you even realizing that you are, right? Because it is asymptomatic. Kind of like, the COVID, that like the COVID virus and everything, the coronavirus, some individuals could be shedding that virus without realizing because they are not showing any symptoms and they would look absolutely fine. And they're not feeling anything, but yet they are shedding a lot of the virus. And it doesn't matter actually whether you're showing symptoms or not in terms of how much virus you could be shedding. Now, they do say, of course, that you could be shedding like a lot of virus, especially when you are showing symptoms, because this is like when you are mostly sick. But again, with certain with certain viruses, etc., even though you are asymptomatic, you could be actually shedding the same amount of virus, okay? And that's what they discovered with the coronavirus. You could be shedding exactly the same load even if you are asymptomatic. And then again, like even if you're not showing any symptoms, the microbe could be active, right, in your tissues. It's just you're not feeling it. And so therefore, well, the host is not going to seek medical attention because they're not going to feel anything. They don't feel anything. So why would they go do like a test, etc.? And as I said, like we refer to these infections as like asymptomatic, or you can refer to it as there's another term, subclinical. Okay. So these are kind of like two terms that could be used interchangeably. Now, how does like a microbe exit out of your body? Therefore, you can go ahead and shed it and start infecting others. Well, coughing and sneezing, okay? Or if you have it, let's say you're carrying it, and then and let's say it's in your blood, and an insect goes ahead and bites you. Now the insect is actually going to carry, right? So say the insect bit you, and now it kind of has some of your blood, right? And so it's carrying that microbe. And then let's say this insect goes and bites another individual. There we go. It's going to go ahead and transmit it to them without without that other person even realizing what just happened. Okay. Removal of blood. Okay, so that's why you have to be very careful when you are dealing with someone else's blood because it could carry, microbes. And you're not aware of it. Feces, right? So stool definitely contains lots of microbes, and that's why you hear a lot of outbreaks of E. coli. because contaminated water, for example, was used in, during like irrigation. And so therefore the produce, let's say lettuce, was contaminated with E. coli. And so an outbreak takes place, and then they would have to go recall, like, these products. So definitely feces. Or let's say someone doesn't have the best hygiene, and so they use the bathroom and they don't wash their hands, and then they go and they cook, etc. Well, there we go. They're going to go ahead and transmit whatever was in their stool, to the food, to the fluids, and then they can, again, this is how some individuals would get it, right? Urine sample. Okay, so you have to be, especially if you're working in the healthcare field, or not even in the healthcare field. I mean, you do not want to, touch someone's blood, or, any body fluid, basically. Okay, so urine is another one. Okay, it will have microbes. You do not want to, you know, um, get in contact, right, with someone else's urine, because again, it could contain definitely microbes. And then, of course, like, open lesions. Um, so again, if someone has, let's say, an open lesion, and let's say it's releasing pus out of it, etc., that can actually, if you touch it, that can actually infect you. So again, you have to be pretty careful. Let's say someone has a wart on their finger. Okay, you do not want to touch someone's wart, especially, again, if it has cuts and it's bleeding. You don't want to touch it, because, that will definitely infect you right away, right on the spot. Okay, if you touch someone's wart, and especially, as I said, if it's bleeding. So, and warts are caused by viruses. And so, that's again another example, of how, the microbes could be transmitted, and how it exits out of your body, and how it can be transmitted to, like, to someone else. Um, and so when a microbe, like, would cause a disease in a host, I mean, the first thing is they have to enter, right? So either they enter through like cuts on your skin, or let's say you ate something, that's bad, and so now it's in your GI tract. You inhaled it, so that's another thing. You can definitely inhale these microbes, and so that's why, um, lots of microbes are found in soil. So you have to really be careful, right? If you are one that likes gardening, etc., you do have to be careful, because the soil contains lots of microbes, and you can definitely, like, as you're, gardening, etc., you could definitely inhale some of these microbes, and you can definitely inhale some of the spores of the fungi, etc. And then, like, through the urogenital, like, tract, etc. So there are, um, different ways, right, of how the bacteria can get into, like, into your system. Or maybe, like, the bacteria is already in your system, like that's part of your biota, but because your immune system has lowered, they're going to turn against you, and they're going to make you sick. So they might be even part of your biota, okay? And then, well, they have to attach, right? So once they're in, they have to attach really firmly, okay? And as I said, your microbiota, especially if they are newcomers, I mean, your microbiota might actually help you out by producing chemicals that would make it hostile for them to, to attach. But um, what are some factors they might have that would help them attach? Like fimbriae, having a slime layer, having a capsule will definitely make it harder for, like, your immune system to go ahead and, and attack them. They might have surface, like, proteins that will again attach to certain receptors on the host cells. Again, viral proteins or viral spikes will go ahead and attach to that specific receptor on the host. And then they would want to avoid your defense system. So they would want to they would want to avoid getting engulfed by the phagocytosis, or sorry, by the phagocytes. The process is called phagocytosis. Okay, so they would want to avoid those phagocytes. They would want to, avoid getting killed. Let's say they do get engulfed. Well, okay, they do get engulfed. Next, well, they have to figure out a way how to not get killed. Okay, because it turns out some of them can actually get engulfed, and then yet they can still stay alive inside of the phagocyte and multiply. Again, that wouldn't be a good thing, of course, because they can multiply and they can start spreading. Um, and then, like, I mean, they can try to avoid like your other, like, immune cells, because I mean, we have other stuff, like we have antibodies, we have B cells, we have T cells. So again, they would try, their best to go ahead and try to evade all of that defense mechanism that you have. And let's say they were successful in evading your immune system, and they are now firmly attached, and they are starting to multiply, and they're kind of starting to colonize your tissues. Well, okay, they're gonna start causing like disease, and if they start releasing, their toxins or their enzymes, we talked about, some examples, and cause like excessive host, like, damage, etc. I mean, then you will be in the disease stage. And we talked about how some of them produce toxins and enzymes, and some of them even change your own DNA, and that can lead to cancer. So we talked about that. And then, of course, like, they would want to, like, they could exit the host, and therefore they could be transmitted to someone else. So we said that when someone sneezes, or through cuts and, blood or urine, etc., or stool, even saliva, okay, will carry the microbes, and all of these are, portals of exits for these microbes. And if they are picked up by someone else, then, that other individual will go ahead and, get sick. But it depends on, you know, um, that other individual. Let's say they did get in contact with like a surface that has microbes on it. It depends how much of that microbe was on the surface, because again, there is that infectious dose that we talked about. And then, of course, it would have to enter into the body, have to attach into the new host, etc. Right? So it would have to go through all of that again so that it can infect someone else. Let's go ahead and take a look at the different stages in the course of an infection. So the first stage is known as the incubation period, and that's basically the time from the initial contact with the microbe, okay? So the first time you contracted that infectious microbe, basically to the time where you're going to start showing symptoms. Um, so it's basically that time period right there, from the first contact with the microbe to the initial, like, appearance of symptoms. During that period, the microbe is multiplying. Okay, so once it kind of made its way through any of the portals of entry that we've discussed, the microbe is going to go ahead and start multiplying, but it's still not causing enough damage to your host tissue to really start showing, symptoms. Um, the period, like, the incubation period will vary from one individual to another, because it will vary depending on how much your immune system is resisting it, right? So again, that's going to vary from one person to another. It's going to vary, depending on the microbe that you've just contracted, how virulent that microbe is. If it's highly virulent, the incubation period is probably going to be much, like, much shorter. If it's not that virulent, again, the incubation period might be, longer. So again, it depends on, the degree of virulence of the microbe. And then it also depends on, as we said, some microbes have specific targets, right? So let's say you've ingested it, right? Let's say you've ingested a certain microbe, but it has to make its way, let's say, to your lungs, okay? So by the time it reaches, right, the target cell, depending on how far that target cell is, the incubation period actually might take longer. So the incubation period can last from several hours to several years. Okay. Usually, though, like majority of infections would range from 2 to 30 days. So that is the incubation period. Let's go ahead and talk about the prodromal stage. The prodromal stage, it's a pretty short period. Okay, it could last from like 1 to 2 days, so it's a pretty short period, and that's when you're going to start showing like the very early signs of symptoms that you're going to start showing. So usually during that stage, you could feel like discomfort, like you're feeling something is not quite right with you. You might start feeling like tired. You might start feeling like your muscles are aching, like an upset stomach, just kind of like a general feeling of, okay, something is not quite right with me. I'm feeling tired. And that's like, during that prodromal stage. Now, during the acute stage, okay, that's when the microbe within you is multiplying at high levels. And it's, showing its greatest virulence, and it's basically kind of causing harm to your tissues, and it's starting to damage your tissues. And so you will definitely, show fever, for example, and or like other signs and symptoms, right? I mean, depending on the microbe, right? I mean, these symptoms might vary, right? The signs and symptoms might vary, of course, from one microbe to another, okay? I'm just giving you some examples like fever, swelling, let's say diarrhea, etc. Those could be certain, signs and symptoms that someone would show. And now the acute period highly varies from one individual to another, and especially also it would vary depending on what is the microbe that you've just contracted. So it's definitely going to vary, and we can't really say, right, like for how long would this acute phase last for. Now, eventually though, right, your immune system is going to go ahead and it's going to try to uh, help you out. Okay, so you have your phagocytes, you have your antibodies, you have your B cells, you got your T cells. I mean, they're all gonna come to help you out, and so you're gonna see that, things are going to start kind of like stabilizing, and then you see how it can kind of start either going down like very steep, like downwards, like effect that you're going to see, or it might kind of like, go down, but it's not that, progressive, like going down. So let's talk about this period right here, and it's called the convalescent period. And that's when, your immune system is working, right? And so you're kind of, your immune system is responding to the microbe. And so therefore, your symptoms and your like the signs and symptoms that you're showing, I mean, it's going to start to decline because you're like, your immune system is helping you out, and you're going to start feeling better, right? You're going to start feeling, okay, I'm starting to feel much better, I'm starting to feel like me again. And again, that's all because of your immune system getting to work. Now, during that time, though, a lot of patients do make a mistake because during that period, again, because your immune system is helping you out and everything, patients, like let's say they are also taking antibiotics. So, of course, you could be taking like, drugs that are helping you out feel better as well. So, let's say someone is taking antibiotics during this period, individuals would stop their antibiotic course. They're like, you know what, I have like, let's say 2 more days to finish my antibiotics, but I feel perfectly fine. So I'm just going to stop my antibiotics. And this is a very huge mistake to do because there could still be pathogens that are still in your system. And so stopping the antibiotics early will again give advantage to these pathogens to go ahead and start multiplying all over again. And so, and again, like, it can also cause antibiotic resistance because let's say there were some pathogens that are now kind of like weak, but you haven't still killed them yet. And let's say they have an antibiotic like resistance. Some of them do, so they're going to go ahead and kind of take advantage, and they're going to start spreading that antibiotic resistance to other bacteria. So this is going to be a problematic phase then that you can enter into. Now you have antibiotic resistance bacteria within you. Now the continuation period, only some infections would actually have this phase. So most of the time, the last phase is the convalescent period, and that's it. We're done. And eventually the person would be pretty well, and you're back to your normal, state. But some infections do have this continuation period, and this is kind of when the microbe just lingers in your system, and so you didn't really get rid of it, so it's just like still in your system, and it can actually stay within you for months or even years, or it could actually stay with you forever. Okay, so some microbes, they can actually stay with you forever, and the patient is going to feel during that stage, the patient is feeling okay, right? And sometimes though, the sometimes like the symptoms themselves will continue. So sometimes like the microbe is gone, but the symptoms do continue. And this is something, for example, that they are witnessing right now with the coronavirus. They're saying that even though, let's say, someone did contract coronavirus, and they're doing tests, and that's it, like they got rid of the virus and everything, but these individuals are still showing long-term symptoms, and that's kind of something problematic that now individuals are facing. So even though there is no virus within them anymore, and that's it, and they're feeling they're still not feeling okay. And so they're still kind of feeling tired. Again, something is wrong. And these are the long-term symptoms that they are saying some individuals face after, contracting the coronavirus. So that would be the continuation, period. Now, if we think about, okay, which of these periods right here we would go ahead and, let's say, shed the microbe that we have and therefore infect others. The highest infection would happen right here during the acute phase. That would be the highest, of like the infection, and therefore like you can highly infect others during this phase. But it turns out actually that you can infect others during any of these periods. So even in the incubation period, when you are not showing any symptoms yet, and the bacteria is still kind of like, has entered into you, and it's kind of like multiplying, but it's still not showing any symptoms, you can go ahead and infect others during the incubation period. You can go ahead and infect others during the prodromal, definitely during the acute phase, the convalescent, even in the continuation period. As I said, with some microbes, and especially with like certain viruses, they can actually linger in your system, and you can go ahead and infect others, without knowing. So during any of these phases, you can go ahead and still infect others. Now, if we take a look at some reservoirs and some transmitters, how do we go ahead and get infected? Well, one is, animals. So like mammals, birds, reptiles, of course humans, but we can go ahead and, let's say if someone gets bit, let's say with a squirrel, and the squirrel is carrying rabies. Well, that's right there, like a direct contact with the animal. If they bit you, if they scratch you, you can go ahead and get it. So even some domestic, animals, cats and dogs, they could be carrying a certain microbe. And so if they scratch you, or even if they lick you, okay, they can go ahead and transmit it to you. Let's say fleas. Okay, so if a flea is carrying like a microbe and it goes ahead and kind of bites you, it can go ahead and transmit it to you this way. Let's say an animal urinated in the water and then you ended up drinking that water. You can get it this way. That's why, again, it's not recommended for you to drink like, water from a lake or a river, etc. Or humans themselves, of course, we can transmit it to others. Someone sneezing and their saliva droplets, landed on you. There we go. You can, you can get it this way. Or if someone again is pretty sick and they just sneezed onto a certain, object or on a surface, and you went ahead and you touched it, there we go. You can get it that way. Um, some human carriers, we call them carriers when they are not really showing any symptoms, right? So they look, they look asymptomatic, okay? They look just fine, but yet they have a microbe and they could be shedding it even though they are asymptomatic. So as we said, with the coronavirus, humans could be carriers. They could absolutely look fine to you, but they are carrying it, and so therefore they could be shedding it. So that's an example right there. Or they could also be, so if you get too close to them, saliva droplets, etc., they can go ahead and transmit it to you this way. Or if um, you know let's say you got into contact with, let's say you're sharing the same bathroom, right? So, via feces, etc., if they didn't wash their hands pretty well, and or again, you're sharing the bathroom, there could be, virus being shed, for example, and so therefore you got into contact with it, and so therefore you got it this way, or via sexual contact. Okay, so HIV virus, for example, again, that individual in front of you might not be showing any symptoms. But the HIV virus, by the way, has a very long incubation period. And so during that time, the person is not showing any symptoms, and yet they are still shedding the virus. So the HIV virus, I think, has like an incubation period of like 10 years. So for like 10 years, someone could be infected with HIV virus and they have no idea. And during that period of time, uh, they could be shedding it to someone else. Some biological vectors. So for example, insects, mosquitoes, etc. They can go ahead and if they are carrying the virus, they will go ahead and pass it on to you. When they go ahead and let's say bite you, etc. They can go ahead and spread it out. Um, let's see. Also, non-living reservoirs. So non-living reservoirs, basically, uh, water, soil. As I said, the soil has so many microbes. So if you are gardening, etc., you're dealing with the soil. A lot of these microbes, you can actually end up inhaling them. Or as I was saying, the soil also has lots of fungus, and so you could be inhaling the spores of the fungus as well. Or the water, air. So as we said, especially bacteria, I mean, they are ubiquitous. They're found everywhere. So if the microbe is in the air and you've inhaled it in the water, or you drank it, etc., these are definitely non-living reservoirs that can go ahead and infect us. Now, the transmission of a microbe could either be direct or indirect. So direct, like you got in contact with an object, like an inanimate object that had the microbe. And so you went ahead and you touched it, and let's say then you touched your eyes or you touched your mouth, and so you had a direct contact, okay? Or it could be indirect, where let's say you got bit by a mosquito and that mosquito was carrying, was carrying like the the microbe, and then this is how you kind of got it. So you didn't really get in direct contact with the microbe. It was, an insect that was carrying it, and then it went ahead and it passed it on to you. So this is kind of like an indirect, but at the end, you went ahead and you got infected. Horizontal transmission, that's when the disease is passed on to you from, let's say, one infected individual to you. So either direct contact, again, you got close to someone and they sneezed on you, and this is how you got it, or sexual contact with someone that was carrying that microbe and you went ahead and you received it this way. Indirect, so again, like you touched an object that was now has the microbe on it, because let's say someone is pretty sick and they put a pen in their mouth and then you ended up using the same pen. There you go, like this is how you can get it. A vector transmission, as I was saying, like an arthropod, insects, bees, these are all like examples of like arthropods, and they are carrying, ticks, and so they are carrying the microbe, and then they go ahead and they bite you. And that's how, if they're harboring that infectious, microbe and they bite you, this is how they can transfer it over to you. Um, or it could be that you ate, let's say you ate some lobsters or you ate crabs, and they were also harboring an infectious agent. And so when you went ahead and you ate these lobsters or crabs, you went ahead and you ingested those microbes inside of you, especially if they were not cooked properly, or if you, like, especially if you ate them raw, right? So if you eat, raw meat, or if you eat like raw crabs, lobsters, again, I'm just giving examples. Of course, there are other things that you can eat raw, but again, these are just some examples. So especially if they haven't been cooked properly, or if they're raw and you go ahead and eat them and they are harboring that infectious microbe, you can get it this way. Vertical transmission, that's when the disease is transmitted basically from a parent to the offspring. So, during labor, okay, or again, like through the milk or the placenta, or even sometimes like the eggs and the sperm, okay, would go ahead and carry certain, viruses, and this is how it can actually be passed on, okay, to your new, like, to the baby. So there was a scientist known as Robert Koch, and Robert Koch came up with a series of steps, um, and we ended up calling those as his, like, his postulates, right? His like suggestions of basically explaining that a certain microbe can cause a specific disease. And he wanted to prove that, because long time ago they actually did not believe that. They did not believe that a certain microbe causes a specific disease. So for example, they didn't believe that Clostridium tetani, for example, is the cause of the tetanus disease. They didn't believe that Clostridium botulinum causes the botulism disease. So he came up with actually a series of steps that would actually prove that a certain microbe can cause a specific disease. And it kind of became the standard then for determining like the causation of these infectious like diseases. So let's go ahead and take a look at the steps that he came up with. So first, right, you would have to isolate the microbe, right, from an infected individual. You would go ahead and you would inoculate a broth, or culture it like on a plate, right? And then you would go ahead and of course do testing to make sure that this is, to make sure first of all, like what is the pathogen that you are dealing with, right? So you have to do some, let's say, genetic testing, or you have to do some biochemical testing, take a look at it under the microscope, etc., so that you can identify, right, the pathogen. And then, step number 3 would be go ahead and take that pathogen and inoculate it, and basically inoculate like a healthy subject, Okay, so what he did was he went ahead and he, let's say for example, you are going to inoculate it into a healthy rat. Now, if this healthy rat went ahead and got sick with the same disease, okay, then this goes ahead and kind of, well, I mean, it's a sign, right, that they have the same one. But of course, what you would have to do to prove it is go ahead, re-isolate the pathogen from that rat that was healthy and now got sick, and take a look at it under the microscope, do the tests, and this is how you can tell that, there you go, the same pathogen that was, like, that infected these individuals right here was also capable of, causing the same disease in another subject. So these were the steps that he came up with to show and to prove to others that, one microbe can cause a specific disease. Now, there are exceptions though to using, like, Koch's postulates, because some infections uh, we have a very hard time actually isolating or and growing in the lab. Okay, so trouble is we can't even culture them in the lab. So how are we going to go ahead and work with these infectious pathogens? And then some infections can only you know cause the disease in animals, and so you cannot test it, like, on something else, or um and some, like, viruses basically, like, have a very limited host range, so you cannot, let's say in the lab we work with mice, we work with, you know other labs work, I mean, mice is the most common thing to work with in the lab, but some labs also use, like, chimpanzees. And again, some viruses, they have a very limited host range, so you cannot just go and say, okay, well, I'm gonna, infect the mice with it and see, what happens. Again, some viruses have a very limited host range, and so, for example, like, human viruses can only cause disease in humans, right? So ethically, you cannot get a human subject and test it on it, okay? Even though, by the way, Koch did that, okay? So Koch went ahead and infected a healthy, young girl to prove that like a certain microbe can cause disease, the same disease in a healthy individual. So that was back then, though, right? Now we cannot do that, right? We cannot take a healthy human subject and do testing on them, okay? So, so therefore, there are exceptions to following Koch's, postulates, okay? So that was it for this week, you guys.